All right, guys, I'm going to show you what I would consider a very basic signal transduction pathway. And you guys are going to explain a model of this type of pathway. This pathway is called a pathway that starts with a G protein linked receptor. What I mean by that is here is a membrane, here is the receptor protein, and these are the G proteins. So these G proteins are linked to this receptor, okay? You're going to have to read signal transduction pathways, and there's different types. But I'm a firm believer if you know the basics of one type of signal transduction pathway, then you should be able to read other ones and figure out what's going on. Okay. It's kind of like a domino effect. This domino hits this domino, which hits this domino, and so on. Okay. Now, there's three parts to a signal transduction pathway. There's reception, there is transduction of the signal, and then the cell ultimately responds. Okay, so cells can communicate. They can communicate via hormones. They can release a hormone, and that hormone can travel to another cell, and that's a signal for that cell to do something for the organism. Okay, so we're going to start with a liver cell receiving a signal. Okay, let me give you a little bit of background of liver cells. After you eat a meal, when you have a lot of glucose in your system, you need to store that glucose because you don't need to use it. And by the way, glucose can damage your cardiovascular system, so you need to remove it from the blood. Liver cells can be activated by insulin, and that causes the liver cells to bring in glucose and to chain that glucose together into a molecule of glycogen. So I've mentioned before that glycogen is a polysaccharide. It's made of several glucose subunits, and animals store their glucose as glycogen molecules in liver cells and also in muscle cells and a few other types of cells. Okay, so we're going to talk about the fight or flight response. You're walking on a trail, you look up, and there's a bear 10 feet from you. You're either going to have to fight the bear or you're going to have to flee. Okay, now you probably know that, or you've heard the term like the adrenaline starts pumping. What does that mean? Well, adrenaline is a protein hormone, and it is made and it is stored on the adrenal glands that sit on top of your kidneys. And when you get scared, those adrenal glands, those cells with adrenaline in them, will release adrenaline, and it moves into your bloodstream, and then it gets delivered to cells eventually, like in the liver. Okay, so here comes adrenaline. And adrenaline is the signaler, it's the ligand, and it binds to this receptor protein. Again, it's a G protein linked receptor. This is the reception of the signal. So the signal has been received. When the ligand binds to the receptor protein, it causes that protein to change shape. And when it changes shape, then it activates one of these G protein subunits, and it breaks off and it moves and attaches onto an enzyme called adenylyl cyclase that is embedded in the membrane of the liver cell. Again, when this subunit binds to adenylyl cyclase, it causes it to change its shape. And after it's had a shape change, it has the ability to bind to ATP. And notice what I'm going to do here. I'm going to right click and I'm going to ungroup those because I have those grouped. Okay. And it takes off two phosphates. So now it's not ATP anymore. This is called AMP. Remember, cyclic AMP, adenosine monophosphate, because it only has one phosphate on it. Okay. Now, cyclic AMP is called the secondary messenger. It is the molecule that is now active inside the cell that can spread this signal, can activate other molecules, basically. So this is what we mean by transduction. It's kind of like the signal is spread to other molecules in the cytoplasm. So let's talk about what CAMP can do. CAMP activates an enzyme, which is called a kinase. And this first enzyme is actually called kinase A. Now, kinase A is a protein. It was made by an unattached ribosome, and they're just in the cytoplasm waiting to be activated because right now it's off. And it's off because it has these two regulatory subunits on it. Those blue circles are the regulatory subunits, and they're keeping the kinase off. So this kinase cannot catalyze any reactions until those regulatory subunits are removed. Well, that's what cyclic AMP can do. It can bind to kinase, and it can remove these regulatory subunits. So we're basically turning that kinase on. Get those out of the way. It's not quite on yet. Move it out of the way. Cyclic AMP also then transfers a phosphate from an ATP in the cytoplasm. There's millions of them. Notice I'm going to ungroup those to kind of show you what happens next. So it takes off a phosphate from ATP and places it on that kinase. Now, this kinase is active. And this kinase has had a shape change when that phosphate group was bound to it. So that is how ATP is used. And that's literally just it. So I know we say that ATP provides energy for reactions. Well, really, you know what it does? It just provides a phosphate. And when the phosphate hooks on to another molecule, that molecule changes its shape. It has a different structure now. And now it can do something that it couldn't do prior. Okay. So now we have this active kinase A. Kinases activate other kinases in the cytoplasm. So here's another kinase. This is kinase B. It's off. It needs to be activated. And kinase A can bind to it. It binds to an ATP molecule, and it can ungroup it here. It can transfer a phosphate group from the ATP onto the kinase, and now I've activated kinase B. Kinase B, it's had a shape change. It can now bind to the next kinase in this signal transduction pathway. It also binds to a molecule of ATP. That's not going to want to move for me. Okay. It will take a phosphate off of ATP, transfer it to that kinase C, and then I have an active complex here, okay? Ultimately, we activate a kinase that can activate this enzyme. Notice I made it a little bit different, okay? So this enzyme is activated by kinase C, and once again, it's activated whenever a phosphate group is transferred from ATP onto it. So now I have an active enzyme, and this enzyme's job is to break down glycogen. So it's going to break off these glucose monomers. And now that they're free, they can leave the cell via a transport protein because they're polar. So they can't cross through the membrane and go right out. They have to leave through a transport protein out into the bloodstream. And now you have glucose that can be delivered to your cells so they can make ATP, because you're either going to have to run from the bear or you have to fight the bear. And you're going to need a lot of energy to do that. Okay. One thing I need to point out is these signal transduction pathways have to be shut off. So if this kinase A stays phosphorylated, it's just going to keep activating kinase B's. And it's kind of like with negative feedback. We have to have a way to shut down these processes when we don't need these processes to happen anymore, right? You don't need to keep dumping glucose into the bloodstream if that bear is, 5 miles away, okay? So note, there are enzymes, and it's their job to dephosphorylate. So they can then hook on to like this complex, like kinase A with this phosphate group. And then to deactivate it, all they do is they remove that phosphate group. And now my kinase A is inactivated. As a matter of fact, it'll change back to its regular shape. And that allows those subunits to bind back onto it, okay? Also note that hormones kind of have a short shelf life. They can eventually break down over time. So therefore the signal is removed from this process, okay? So again, just note that we have to be able to start these series of reactions, but we also have to have a way to shut them down. Oh, and before I forget, the response part. So reception, transduction, and then the final response is this cell releasing that glucose into the bloodstream.